Welcome to the Daily Planet Theater, the SECU Daily Planet Theater, and this is our giant iconic globe outside, and inside we sometimes play you videos in here, and sometimes we introduce you to cool scientists. And in this case, it's one of our scientists, but she's not here today. She's far away. I don't even know how far away. You're in the Bahamas, right?
Yes, I'm on Abaco Island.
And you're on an island, is that right? Let me introduce you to, this is Dr. Julia Stevens. Hello, Julia.
Hello.
Good to see you. How long have you been there?
I've been here since Tuesday.
Since Tuesday. And where exactly are you? You're on an island?
Yeah, so I'm on an island in northern Bahamas. It's Abaco Island, and I'm actually in a town called Sandy Point. So it's the southernmost region of the island. And we are staying at the Bahamas Marine Mammal Research Organization. Right now I'm in their offices, and we're staying in their home and doing our research from there.
Right, you told me that you're staying at the home of some whale researchers. So they're basically whale researchers, which is not what you are.
Not at all.
And are you there, is this related to their research?
No, so this is related to knowing the people that were the other scientists on this island. So there's an organization here on Abaco called Friends of the Environment. And so it's a good connection for all of the researchers working on the island. And I am here with Layman's Lab from NC State. And they've been working on this island for about 10 years. And so they've made a lot of good connections, and the whale researchers opened their home for us staying here this week.
So let me ask you about yourself. So I know that you've done a lot of work with microbes. You work in our Genomics lab. How do you describe yourself? What kind of scientist are you? Are you a geneticist? Are you, what are you?
All right, so I'm a microbial ecologist.
Ah, okay, a microbial ecologist, which is what?
Yeah. So what it is I study how microbes live in the natural world and what they are doing there. And so a lot of times when we think about microbes, so bacteria, fungus, we think about the bad ones that make us sick. But really, that's a very, very small percentage of the microorganisms in the world. And so most of the microorganisms we rely on for environmental functioning, for health, to be able to breathe air, all came from microbes. And so they're really important in the environment and the ecosystems. And so I study the microbes that live on plants and animals and how they help them.
And that's what you're doing there, right? So you're there to study the microbes in the system there. So maybe you could describe that. What system is this? What are you actually trying to figure out, and how are you doing it?
Absolutely. So I'm here for two different projects. And so the first one is looking at the microbes that live in the sponge called Yersinia felix, and its common name is the stink sponge because it smells really, really bad.
Does it really?
Yeah. And so previous research has shown that as the water goes into the sponge and as it's pumped out, the nutrients and the chemicals in that water changes. And so there are two roles that can happen, is that as the water goes through the sponge and is pumped out, it can either add nutrients to the water as it's going through, or it can take up nutrients into the sponge, and so the water coming out of the sponge has fewer nutrients. And so that's hard to explain by just the sponge alone. And sponges, as we like to call them, are just big bags of microbes. So they have whole big system of microbes that live inside of them. And so I'm here to collect sections of that sponge and characterize the bacteria that are living in them and look at how they're changing the nitrogen in the system. And so nitrogen is a big indicator for pollution. So our chemical fertilizers are mostly nitrogen. And so as development happens and we're adding fertilizer nutrients to the system, this could have really important implications of how the reefs that are where that water ultimately ends up are responding to pollution.
So the nitrogen levels can be bad, like too much nitrogen in the water. What happens if there's too much nitrogen in the water?
Yeah, so these waters are, we call them oligotrophic, which means naturally there are very few nutrients in the water. And so the systems here are adapted to trying to suck up and retain as much of the nutrients as possible whenever they're introduced. But what happens is when we introduce too many nutrients, and so you have this influx of nutrients happening, is it knocks the whole thing off balance. And so algae really likes the nitrogen. And so what happens is that the algae can grow faster than the sponges or the corals, and it can take over everything and smother the other coral reef organisms. And then when that happens, then you lose a lot of the fish diversity because the fish rely on the sponges and the corals that are there. And so basically what's happening is, like in your lawn, when you add fertilizer, you get this nice monoculture of grass. That's happening in these reefs, except it's harmful algae that's growing.
Okay, so I think you just answered the thing I was about to ask you. Was because you're studying something on the microscopic level that has effects that are visible on a big macroscopic level. And I was going to say, so are you there in that particular area because of observations that were already made, that something's going on there that's not good? And that's what you basically just said, isn't it? That you're already seeing something problematic, and so you're trying to get at the root of it?
Exactly. So here, this is actually, for the most part, a healthy system. And so if we understand how the healthy systems work, and that we are getting to the maximum capacity that these organisms are able to process, and then that can inform future conservation in these systems that are still really healthy. And so what we can do is, if we find the level where the nutrients are harming the environment, then that we can put a cutoff there.
Okay, so there are other areas where this harmful stuff is already seen to be happening. You're studying a place that's healthy so that you can understand what a healthy system looks like on that microbial level.
Exactly.
And then and then you look at the unhealthy ones and you see the difference and see how what you can do to make them healthy again.
Exactly. So what we're doing is we're collecting the baseline data. So we need to understand how the healthy system works before we can try to fix a bad system. And so we're here collecting for some preliminary data so that we can start to inform bigger projects and develop bigger projects on this information.
Right. That's an interesting idea that I've heard, like the basic idea there of what you just said. I've heard this discussed in terms of human health, that a lot of times, and it could be mental or physical health, that we go to doctors when there's a problem, and we study situations that go wrong, but there's a lot to be gained from studying the healthy animal or the healthy ecosystem.
Exactly.
Because, how can you study the unhealthy one without having that, you said, a baseline to compare it to? Very
exactly.
interesting.
And so that's what a lot of ecologists do, is that we study how the system works in the ideal setting, because then we can figure out what different effects are doing to the system. If we don't know how it should work, how are we going to say, oh, well, this is due to pollution or climate change or X, Y, or Z?
And as is often the case in science, you mentioned that you're doing this in partnership with other groups and some people at NC State. And in a way, you're like coming in to do what you do well on their research project. But I know that you have research projects of your own. And so you mentioned you're there for two reasons. And so the other reason has to do with laying some groundwork for your own research, right?
Exactly. So my research is on, as part of some of my research is on the microbes that live on lionfish. And so I did an assessment here in the Bahamas 4 years ago, looking at the microbes that live on the skin of the lionfish that actually help them fight disease. So these bacteria that live on the lionfish skin can inhibit the growth of fish pathogens. And so it's keeping the lionfish healthy. And so what I am doing now is I want to develop a project and looking for the source of those microbes. And so actually our day yesterday was traveling around to different reefs just so I could get an idea of the system here. And then this morning I was working on developing a research program here in Abaco that will continue with Craig Layman's lab. So I was brought in for my microbial expertise for the sponge project, and now I'm reciprocating by bringing them in for their access to these reefs, knowledge of the system.
And maybe tell us a little something about the lionfish. Because
Yeah.
I know that I've heard you talk about it before, but for someone who doesn't know why the lionfish, tell us something basic about the lionfish and why you're studying it.
Yeah, so lionfish are one of the worst marine invasive species in recorded history. So
What makes
lionfish
them, like, what does that even, and tell us what that even means. Like, what makes it such a, one of the worst invasive species?
So one of the worst is its growth was extremely rapid. So it's population growth, not the individual lionfish growth. So populations exploded all along the U.S. eastern seaboard, throughout the Caribbean Sea and the Gulf of Mexico. So a very extensive geographic range. And on individual reefs, the population, you would get 40 times more lionfish on any given reef here compared to its native range in the Indian and Pacific Oceans. And lionfish eat everything. Anything you give it to eat, it will eat. And so that's bad because what happens is it likes the smaller fish, and so it's eating all the babies off of these reefs. And so if you eat all the babies, then there's nothing to get big and how to keep the fish population healthy. And so lionfish were introduced in Florida through the aquarium trade. So they're beautiful fish. You can see them in the tanks here at the museum.
And we have one on the screen as well.
Oh, you have
So
perfect.
So yeah, they're very beautiful. I understand now. The spines are they're as beautiful as they are. They're to be avoided because those spines are dangerous, right?
Yes, they are venomous. So on the spines along the back of the lionfish, that the skin over it is actually covering a spine that it's sort of looks like a hypodermic needle. So the needle you would get like your flu shot with. And what it does is when it punctures, it injects a venom into whatever it hits.
Well, that's probably pretty good protection for them. Is that one of the reasons why they don't have a lot of predators, or they have predators where they're native, but since they're spreading into new areas, there aren't really predators for them there? Or what can deal with those spines and eat a lionfish anyway?
So we don't know. We don't know in their invaded range. We don't know in their native range. We don't know what eats them. We don't know what controls them in the Pacific. So it could be that there is something eating them and they're just not in high enough abundance. Not a lot of research goes on there on the lionfish. It could be that there are other species of lionfish that are competing with the Pteris volitans. So that's the species that's on the screen. And so that's the one that we have here. It could be a disease or a parasite that's controlling it. We don't know.
So why are you studying? Okay, so why is your research not even about the lionfish itself, but about the microbes on the lionfish?
Yeah, so we know that all plants and animals have some sort of microbiome, and they rely on the microbiome to different extents. So some completely rely on their microbes, and some it just helps them stay healthier. And with invasive species, there are a few different patterns of the microbes that are associated with them that we see. So the first is that when an invasive organism is introduced into a new range, it can introduce a disease. Or what can happen is that when the species are transported, they can lose the bad pathogens that are harming them, but they keep the beneficials. And we see that pattern, so losing the pathogens and keeping your beneficial symbiotic bacteria. We see that when invasive species come through the ornamental trade, so ornamental plants, aquarium fish, pets, because what happens is in the native range, they're picking the healthiest and the strongest, and those are the ones that are being sold through the trade. And so that's how lionfish got here. And so it could be that they picked the healthiest looking lionfish from the reefs, and those get sent to the US to be put into people's aquaria. And then we don't know the exact time. It was about the mid-'80s that they were introduced, and they're from, most likely from aquarium releases. It could be that there were eggs in aquarium water that were flushed, but it was a very small introduction in Florida. And so it only came from a few individuals. And so what I was interested in is, do we see that pattern of losing the pathogens and keeping the beneficial microbes?
So on this particular trip, you're just sort of doing some preliminary, you're not actually working on that, but you're scoping out places that you can come study when you come back on a trip dedicated to the lionfish?
Exactly, exactly. So, and that's how a lot of field research goes. You have money and funding for a project that you're following up on and getting more data, but then you can use that opportunity to develop other projects at the same time. And so that's how we keep research going and can afford to come into the field, is that we have these projects that are funded, that we're going and we know what we're doing already, but then we take the opportunity to understand the system to develop more questions.
And then how do you spend your day there? What are you doing? There's an image, you sent us some photos, including like showing how you collect the sponges, but so is this what you're doing? Are you putting on wetsuits and going diving every day?
Yeah.
How are you actually spending your time?
Yeah, so what we're doing is we, each morning we have a van that's loaded with our scuba tanks. We have a small inflatable boat in the van, and all of our collecting materials. And so we go out to these reefs and we find these sponges. And so we're looking for one species, so we're controlling that we're just looking at the same sponge species. And so what happens is that we inject the water around the sponge with this fluorescein dye. It's bright, bright yellow. And we're checking to see if the sponge is active, if it's pumping. And so what happens is you put the dye around the edge, and I'll have to do a talk when I get back because I have some pictures of this.
Cool.
And so if it's pumping, then after a few seconds, you'll see the dye come up through the sponge. And so then we know if it's pumping. And so then what we do is we're taking, with a syringe, we're pulling back on pulling the water that's the inflow, the in-current water. And then we're also collecting the water that's coming out of the sponge.
Okay.
And so we're getting the water as it goes in and the water as it comes out. And so we can test the difference in the chemicals and the nutrients between those two. And then once we do that, I take snips, so underwater scissors, and I cut a portion of the sponge. And so I cut the portion of the sponge and put it in a baggie. And then as soon as we get on land, then we're taking our samples, we're putting them in preservative, and then we get them to a freezer as quickly as possible. And so, yeah, so that's done in scuba, using scuba. And we also yesterday was mostly on snorkel because we were scouting sites. And so we didn't have to be down on it. We could just see it. And so we were only in 10 feet of water at that point.
Are you having fun there? Are you enjoying doing this part of the work? Is this I know a lot of times people think of scientists spending times in laboratories but all our scientists tend to spend part of the year going out like this, we say in the field. And yeah, it sounds like you're having fun. You look very happy and you're diving. You like to dive. You like to be in the water, don't you? So
yeah.
It makes
field work is my favorite part, and it's sad because it's always the shortest part because the lab and then the data analysis both take forever. But
so
the field.
You've been there a week or so, and how much longer do you have there?
Not long. I'll be home on Monday.
Okay.
Yeah,
well, thanks for taking the time to talk to us. I don't know if anyone has a question. Oh, would you like to come on up here a sec? Let's take one question here from an audience member. What's your name?
Sharon.
Sharon, what would you like to ask?
Hello,
So,
Sharon.
Julia, you were saying about the algae fields that they can see it. Is that something that we're seeing in North Carolina? Is that why we're doing this? Because we see unhealthy water here?
So this is funded by a private donation, and so this is being done in the Caribbean because this is where this system is set up. But we can do this off the coast of North Carolina. I actually haven't done, as sad as it is to say, living in Raleigh, I haven't done any work off the coast of North Carolina. And so I think it would be really interesting to take these questions and do our more temperate environment, so that's in North Carolina, compared to this tropical environment. And I think that that would be an important thing to test. So the actual system is a lot different. So we're not actually on the ocean. We are in a bay that's protected. And so we have these very different reefs than in North Carolina. But yeah, it would be really interesting to look at.
So it's a
the
good
comparison.
question, but we don't know yet, right?
Yeah.
Excellent. Any other questions from anyone here? All right. Well, Julia, thanks for taking the time to get out of the water and talk to us a little bit about it. Maybe next time we should Skype from the water. I don't know if that's possible. But good luck, and we'll see you back here, and I'm sure you'll have lots of cool pictures, and you can come tell us all about this in person.
Will do. I'll see you next week.
See you next week. Thanks. Bye.
Bye.
All right. She works